Ladies, today we're going to talk about viruses. We're going to take notes on what viruses are, the parts of a virus, their life cycles, how they work. Feel free to stop the video at any time to take notes or to backtrack if I'm going too fast for you. So we're going to start with parts of a virus. And you can see the three pictures of the virus, of different viruses here. We have a T4 bacteriophage first, the tobacco mosaic virus, and then an influenza or a flu virus. And one thing that you're going to see that's in common with all of them is every single one of them have genetic material. Now the bacteriophage has DNA, the flu has RNA, so it may be different genetic material, but they all have genetic material. The other thing that all viruses have is a protein coat. So some of them, they've called them capsid proteins or capsids or surface proteins. In the bacteriophage, it's like the head and the sheath tail. But essentially with the protein coat, it's an outermost coating over the virus, and it impersonates proteins found in the body. So what happens is the proteins on the virus fool the body into thinking that it's a friend. And then the body's like, sure, come on in. We're happy to see you. And a body cell actually lets the virus inside of it. So this allows the virus to sneak into the body. So we're going to get into virus life cycles first. But a lot of people are always like, oh, is a virus living? Is a virus non-living? So technically, a virus is a non-living thing. Viruses can only reproduce by infecting living cells. And for that reason, they're considered non-living instead of actually living and actually reproducing. So let's get into the life cycles. So viruses reproduce in two different life cycles. There's either the lytic cycle or the lysogenic cycle. And we're going to go over the lytic cycle and the lysogenic cycle in detail. So here's the lytic cycle. I have much better pictures than this. But in the lytic cycle, viruses reproduce immediately. So you can see in that picture, the T4 bacteriophage, it's the blue thing that kind of looks like a spider, is infecting a bacteria. So even bacteria can get infected from viruses. And the bacteria is the little orange thing. So the bacteriophage ends up the proteins end up sitting on top of the bacteria, and that ends up tricking the bacteria into thinking it's a friend. So then the virus inserts its DNA, and then what happens is the bacteria gets confused and starts reading the viral DNA, and the viral DNA says, turn into a virus-making factory, and so it does, until the whole cell explodes with new viruses. So let's look at that step by step. So step by step, the steps of the lytic cycle. First, the virus attaches to the host cell because the host is fooled by the protein coat. The next thing that happens is the virus inserts DNA into the host cell, or in the case of an RNA or retrovirus, it's going to insert RNA into the host cell. The cell then reads the viral DNA and it tells it to make virus parts. So basically, the cell gets confused, it starts reading the virus DNA instead, and boom, it starts making virus parts. The virus parts are then assembled into a bunch of different viruses, and then the viruses break free from the cell to infect more cells. And the flu is a great example of this. If you have the flu, one cell can infect one of your throat cells, for instance, or one flu virus, one viral cell can inject or infect one of your cells. And out of that one cell, you'll get about 10,000 new viruses. So that's a lot. Okay, so now we're coming to the picture, a better picture really of the lytic cycle. So you've got the green cycle we're going to talk about later. So we're on the blue circle here. So again, remember that the scary spider looking thing is the T4 bacteriophage, which is a virus. And then the yellow oval looking thing is a bacterium. So in our first step here, you have the virus injects DNA into the bacterium, and you can see the little purple stuff coming out of the virus. It looks pretty gross. Okay, so once that happens, Then the viral genes are transcribed by the host cell. So it starts reading that DNA. Then once it reads it, the bacteria makes new viral proteins and new nucleic acids. So it's all the pieces of the virus together. Then, kind of like an assembly line, the proteins and nucleic acids are assembled to make a brand new virus, or rather brand new viruses, like 10,000. Then the viral enzymes lyse. Hey, remember lyse, kill, break, explode? So the viral enzymes lyse the bacterium's cell wall and the new viruses escape. And like I said, with the flu, it's about 10,000 per cell. So really, the job of the immune system is to battle a virus or a bacterium, but let's talk about viruses since we're talking about that today. You have the cell-mediated and the humoral response. The cell-mediated response's job is to sit there and be like, okay, I need to take out with a T cell one of these infected cells. The humoral response is to take out the B cells make antibodies which take out free-floating viruses. So together with taking out cells that are already infected and free-floating viruses, it's like a 1-2 punch and your virus is eliminated. Now let's take a look at the lysogenic cycle. In the lysogenic cycle, viruses are dormant. So viruses insert DNA or RNA, which becomes incorporated into the cells. And it becomes incorporated into the cell's DNA. So it kind of lays hidden there. Those cells containing viral DNA can then be replicated. And the cell goes around carrying on its normal functions, but it has that hidden viral DNA. And every now and then that viral DNA can break free and start becoming a virus again. And then you get like an outbreak or a flare. So this is really common with oral herpes, which you are probably like, oh, herpes, but just cold sores on the outside of your mouth. That's oral herpes or herpes simplex 1. So the reason why that works is maybe you'll get a breakout when you're stressed or when you've had certain foods or something. And again, it's just something in your body triggers that release of the DNA that lies dormant to then become undormant. So here's a picture of the lysogenic. So we looked at the lytic, which was blue before. So now we're on the green side, the lysogenic side. So again, the virus injects DNA into the bacterium. This is the same process each time. But this time, the viral DNA inserts itself into the bacterial chromosome. Now, they're circles because bacteria have circular DNA. How cool is that? So if you can see this the arrow there that says prophage is pointing to that purple piece of DNA that is viral DNA that is now incorporated into the bacterial DNA. Okay, the prophage may replicate with the bacterium for many generations. Notice we make a couple of cells. Then for whatever reason, the prophage, the viral DNA, can exit the bacterium chromosome, and then it just starts going straight into the lytic cycle. So something that's lysogenic can stay dormant for your entire life, or the prophage can exit and it can enter a lytic phase. And what's even more annoying about things like herpes simplex 1 is then the viral DNA can hide back in the DNA and lie dormant again. Okay, so this is just, I like this picture to show kind of the cyclic nature of the lytic and the lysogenic cycle. The fact that you have this idea that the lytic is the one that is really exploding out the viruses, right? But the lysogenic cycle just keeps going dormant and dormant. Here's the yellow in this case is the viral DNA and the purple is the bacterial's DNA. And you can see here how it's being incorporated, or I guess here, how it's being incorporated. So it's being incorporated into the bacterial's DNA there, and you can see now you're getting a bunch of cells that are coming out that have hidden DNA in them, and at any point these cells can decide for whatever reason they want to bust out and then start heading into the lytic cycle. So which is worse? Is it lytic? Is it lysogenic? Let's take these step by step. So in the lytic cycle, the virus infects one cell to start off with, and the infection immediately spreads throughout the body. That's not very good. In the lysogenic cycle, the virus inserts its DNA and it doesn't cause infection initially. Great, hidden infection that doesn't really get me sick. I'm okay with that. However, if a lysogenic cycle suddenly becomes lytic, now you have many cells starting with the infection. This causes so much more damage, right? So instead of one cell starting with the infection, now you have 10,000 cells starting with the infection. And one cell making 10,000 viruses, not good, but 10,000 cells making 10,000 viruses each, really, really, really not good. So overall, in order of like best to worst, lysogenic is by itself is probably the best, then lytic, then a lysogenic cycle that turns lytic is the absolute worst. All right, ladies, so that was just a brief introduction to viruses. We're going to read more about viruses and answer some more questions on Monday, but for today, you're done. Monday is a work day, and I will see you Tuesday. Have a great weekend, ladies. Bye.